Okay, so thank you, everyone. Thank you for the nice introduction, and thank you for inviting me to talk a little bit about the future of concurrent TMS-fMRI. So first of all, some housekeeping. So I do have a license agreement with the Medical University of Vienna for that course that I did during my PhD, and we also have now a patent granted for about this the RFE GCAP. So before I go into my talk, I want to put out this kind of philosophical question. It's going to be very dark now. This philosophical question, what is, we all build technologies and methods. So what does really a technology need to be transformed into a scientific tool? Okay. So this question, I had it in my mind and I said, what do I really need to accomplish so that we can say that it's a scientific tool? So I found out these 6 elements, and I hope you agree with that. So first of all, it has to be an extension of human perception. Then it has to deliver we have to be able to measure with some kind of precision. Then it has to be reliable and reproducible. And then it has to give us some kind of possibilities to do some standardization and calibration. I know that with fMRI, that's not so easy, but maybe more from the TMS side. Then it has to be accessible and usable. And then it has to be validated. So we have to be sure that what we are measuring is not some artifacts, or that we are like changing something that we want to measure. So with that in mind, I mean, first of all, keep these icons in mind because they will be showing up during the talk, so that you know that each of that means that kind of characteristic. So this is the outline of my talk. Of course, we cannot talk about the future without talking about the past and the present. So, but I do believe that the RFCAP, this is this specialized hardware that I've been building during my K99, is gonna give the full brain concurrent TMS-fMRI some capabilities to bring this tool into a more scientific tool. So then I will talk about other methods to improve, for example, calibration and reproducibility, and then I will sum up. So let's start with the past. So who of you are here familiar with TMS, or maybe is there anyone in the audience that has no idea what TMS means? Should I do a kind of introduction? That's fine. Okay. So the first time that the concurrent TMS-fMRI experiment was done was at the Medical University of South Carolina. It was in the group of Mark George, and the leader of that investigation was Bonin. And they just decided, to put together into a 1.5 Tesla system. They kind of modified one of these TMS coils to make it MR safe, and they put it inside of the big gauge coil. They use what is called an interleaf acquisition. That means they put a pulse, stimulated the brain, and then they acquire the images, and there's in a blockwise, so that they could have some kind of statistical significance. And this is what they show there. I mean, this is the figure there. So you can see that under the TMS coil, there's some activation. Okay, you can see also other things like a lot of distortions and whatever, but I mean, it was very brave to go and do this experiment just once, because the TMS produces a very high magnetic field, and they were afraid that that was going to explode or break or whatever. So we have to give them that, and there was a lot of interest from the community to do these kind of experiments, because what the field wanted is to change this idea of we are studying brain function just by observing something in this kind of with the fMRI, from we activate some parts of the brain in a very defined and controlled way, like with the TMS coil, and then we observe. So it's a totally different change in the paradigm. So the conclusions of that experiment was that concurrent TMS stimulation and echoplanar ball fMRI imaging is possible. So we can say that the new technology was born, and it's definitely an extension of the human perception. So in that first publication, they listed all these issues that they already, realized. And these, they were all described in this first publication. And the years after that, and this was at the end of the 90s, in the past century, the years after that, many researchers work on, taking care of all these little things that they found to make or to push the technology further. So I'm going to start, for example, with the relative timing of the EPI acquisition and the TMS stimulation. They found out how important that is. There is for the people that are new to the field, there is a seminal paper from Bestman et al. In 2003, where he showed, what happens if I place my TMS when I'm doing the excitation or maybe the gradients and so on. So this is some work that we presented at the ISMRN. We placed the TMS exactly in different parts of the sequence also. And we could show, that especially when you have the gradients, very close to the gradients, this is going to have this terrible artifact. So you are going to have horrible signal drops out. From that studies, and also, it has been shown that if you do like an interleaf acquisition where you have a little gap or you space it, that you have like about 100 milliseconds before the gradient, after the gradients and everything should be fine. There are other methods. You can also live with the idea that one of your volumes are going to be affected. Maybe it's only one volume, maybe it's two volumes, it depends. So you could also do some interpolations. But if you don't want to have any effects, just have a little gap, wait a little bit after the TMS pulses, and then you are fine. So this contributed, as I said, to this validation. Then other issues that they found out is that because they placed the TMS coil directly through the door, that the RF noise was much higher. So what they find out is that the best idea to control that is to have what is called an RF filter. And this is the way this has been adopted by the vendors, the solution. So they place an RF filter here. Everything is outside outside of the RF shield room. So the stimulator is here. You have this cable that is going to be connected to the RF filter, and then it goes through the waveguide to the TMS coil. So then we can control the RF noise levels. Then for the leakage currents, the same. Baskov has a very nice paper about that. They realized that even when we were not pulsing the TMS, we were seeing some effects from the TMS, from some leakage currents. So he modeled those currents and he proposed a compensation method, which has been adopted by the vendors. And now we have this leakage current system in our stimulator, so that is really minimized. So as I said, this also contributed to push the validation. Then we have other effects in the scanner, of course, that are more difficult to control, for example, the torque. So the TMS is producing a very high magnetic field, and that's inside of another very high magnetic field, and they are not aligned. That's the biggest problem. So when you pass the TMS, it's going to try to move to align with the B0. So in order to avoid that, we need some kind of fixation system. Moisa proposed something in 2009, which actually, this system is actually the precursor of the commercial available system that are now, it's not perfect, but it helped a lot, and it helped a lot for the, for the researchers to keep doing these experiments. Then the topic of the sound. The sound is going to be a big confound, and there's not much that has been done there. Petrushev has been working on that, but not on the MR-safe TMS coils. So what I really could recommend most of the times is to have some kind of absorbing material lining in the bore, so at least we can control the echo that it produces, because these loud noises are inside of the bore, and that produces a lot of, most of the sensitive or the sensation that we have with the sound is of this echo, this doing everything in the bore. So all these methods also have helped a little bit with the usability and reproducibility. So now the B0 effects. So we have this big coil that is a big chunk of copper with some plastics on the top of your head. So I'm going to have an effect on the B0 homogeneity. That means we are going to have geometrical distortions. So geometrical distortions per se, we can correct them, but the biggest problem is when you have signal drop-offs. And that was the case when we were using the Birkage coils, because that was you didn't have any kind of speeding up the acquisition. So that was why right now using faster acquisition methods, parallel imaging, can help a lot because it reduces these effects, but that requires a lot of SNR, and that is something that they didn't have at the beginning of the 2000s. So that was important to be aware that there were going to be distortions that could be that you don't have any signal at all there. So now the present. How does it look like now? What is the status of the current TMS-fMRI? So if you want to conduct these experiments, what you are going to have is a TMS-fMRI system. First of all, you have to decide which kind of RF coil you are going to use. You can use either a Birkage coil that is shown here to have full brain coverage, or you can use these surface coils. That's the disc that I developed in Vienna. You can have one with a TMS, an extended one for to have extended field of view. But you have to decide in which direction. And that RF coil is going to define your measurability, your precision. Then we have the MR safe coil. Then we have also the TMS, the fixation system here. And these kind of surface coils, the beauty of those ones were that they allowed for the first time to use neuronavigation systems. That was not possible really with these Birkage coils. So that opened up a lot of possibilities to the investigators to study other motor besides the motor area, other areas, to put it on the DLPFC and be some kind of able to reproduce those measurements and do it. Then, of course, then we have for the stimulator, the RF filter, the things that I discussed before, the synchronization for the TMS, that's the one that are responsible for the validation of your technology. So, but still, one of the biggest challenges, as I said before, is which RF coil you're going to use. So we cannot use the typical 32 or the 64 channel coil. There is no room for the TMS. The TMS is pretty bulky, so you cannot use it with these ones. So the other possibilities, as I said before, the Birkage coils, you can also, there are some people using these spine coils surrounded, or other possibilities like these receive coils on the sides, and they give them different names, always related with taco or sandwich, always related to food. I don't know why, but that's how the community, some are more focused on grappa or something like that, and the others in food. But none of these possibilities that we have, these RF coils, really delivers you the full brain coverage with high sensitivity and high parallel imaging capabilities, and the use also of the neuronavigation to have it more reproducible. So where do we stand really right now with concurrent TMS-fMRI, with these kind of systems that we have? So there's no doubt that it's an extension of human perception, that's for sure. And I am, giving like notes or to each of these characteristics. So I think the extension of human perception receives a 10. Then for measurability and precision, I don't know, do you want to give a guess? What do you think we are at? Or maybe the people that are more involved in the field. Kenny? Apo? Okay, I will say we are about in a 6. It's not great, but it's, it's doable. You get some kind of precision. Then in terms of reliability and reproducibility, that's where it doesn't pass, really. It's very difficult to replicate it. People get very, very different results. There's not a lot of confidence in all the results. Then in terms of standardization and calibration, it's even worse. First of all, because fMRI is not so great, the calibrated or standardized, but also the TMS, we are still talking about resting motor threshold, percentage of resting motor threshold. What does it mean? It's not really calibrated at all. Then in terms of accessibility and usability, it's also still very challenging to do these experiments. People that do it are very they spend hours trying to position the coil, and the coil moves, or the person stops the scanner, or something happens, and then the time is gone. And then in terms of validation, I think the preceding work that has been done was pretty enough. So I think we can say that it's kind of a pass 6. So now let's see, where are we going? Where I see the future of the concurrent TMS-fMRI. So when I proposed the RF cap, actually the first time, it was like a cap where you can just put the cap and you will be in the scanner freely. And then I realized, okay, I need a more realizable, more real, how would I say it? It cannot be so easy. So I have to define it in 2 parts. Like the first one in the flexible part, which is what I had in mind, this neoprene cap. And then I had in mind these flexible PCBs that I could have on the to connect them with the RF elements. The RF elements that I'm using is the technology, the self-resonant coaxial cables. And then we will have a rigid part, which first of all will help with fixation. We cannot leave the head just hanging around in the MR. Unfortunately, we are still not there. But it will also this rigid part will also host the preamplifiers, which is usually the most expensive part of an RF coil. And those will be placed here. The preamplifiers are not shown, but just the PCBs that we place them. Then also all the host interface cables, and it will host also the interface cables, the electronics to the scanner under the base. So this orange base will host all that electronics so that everything is free here, so that we don't have any hindrances positioning the TMS coil. So this is the final layout. It has 26 RF channels. I'm talking about an RF coil. It receives only the signal. So it's not stimulating anything. It's just to capture the fMRI. Okay. And this kind of setup really achieves our goal. More nice pictures from the flexible part. This is the neoprene cap with all those TMS, with all these RF elements. A little bit of photo of those flexible PCBs with the smallest elements I could find. And I put a lot of effort and an emphasis in making it very, very thin. So you can see that that is barely 2 millimeters. And the reason for that is that the TMS, if you are far from the head, because we are stimulating the brain using the magnetic field, the magnetic field it decays very fast. If we are like 3, 4, 5, 1 centimeter away from the brain, we are not going to stimulate the brain at all. I mean, not from the brain, from the scalp. So that's the reason why I put so much emphasis on making it very thin. And that's one of the things that many researchers or developers didn't understand why it was important, why it has to be so thin. Then I added some epoxy on the cables to make it more robust, because of course you are putting this up and down or something, so it has to be a little bit more robust. Then the rigid part shown here, you see all that electronic here. We are using the direct connect, so you place this in the patient table, and then the person can have the neoprene cap, can be, we can do some experiments outside of the scanner or something, and then it comes in, and then we connected everything with the those connectors. So this is how the full system looks like. So this is the TMS coil. As I said, it's a very beefy coil, but you can see that our setup allows to position it everywhere. We are a little bit limited on the occipital part, but the rest of the brain is really open. Of course, we don't do the experiments directly like this. We have this cover. The final RF cap has this cover so that the people are not picking into the electronics, of course. So with this setup, we will see that we are improving the usability, because it's easier now, the positioning of the TMS coil, and especially the reproducibility. So now let me share with you the assessment of the RF cap performance. So I'm going to be talking about SNR maps, signal to noise ratio. So I'm going to compare this with existing setups. And we did all these measurements on a phantom, or on our red phantoms. So the first is with the beer cage coil. This beer cage coil is the one transmit receive coil that, it was here where people tried to do concurrent TMS-fMRI like 10 years ago and didn't go so well. So this is the SNR that you achieve with that beer cage coil. And then we compare it also with the 2 times the surface coils, the disc coils, where you have one on that side where you want to stimulate and the other one in the contralateral side. It does boost the sensitivity, of course, but the biggest problem of that setup is very difficult to reproduce, because there are some parts of the brain that you are not going to be you're not going to have a great SNR, definitely. And this is the SNR, or the sensitivity map on the RF cap. So you can see that we are boosting the sensitivity by a lot. So the RF cap has 4 times more SNR than at the center of the brain compared to the beer cage coil. And on the cortex, we are 10 times to 14 times more SNR. We also have a lot of advantages, compared to the 2 discs, because we are covering the whole brain, really, and in the places where and another thing that I will mention later is the thickness also of our cap. But now sorry? You mean that is the bottom? Oh, that's the bottom. That is the bottom. Yeah, that is the bottom. The top is here. Oops. That is the top, the bottom. And that is below. So now, as I said, the RF cap is going to improve the measurability and the precision. Then we did also compare our cap with commercial coils, with the 20-channel coil and the 30-channel coil. We're kind of between. It is true that we have less SNR in the center of the brain. I believe mostly because we don't have those elements, those eye elements and those elements here more in the front part. An idea to have for the future versions. But still, I want to emphasize we cannot do TMS-fMRI with the commercial RF coils, with the 20-channel coil and the 32-channel coil. Now I want to share with you the performance in terms of parallel imaging. So maybe you are not aware, but the G factor shows how it's going to be affected your SNR when you start to speed up your acquisition. So if it's red, means we are not going to decrease the SNR too much, but if it starts to be yellow and blue, it's going to be a little bit worse. So there was a moment where it's just not going to be good enough to do the experiments. So we were very surprised with the very low correlation matrix that we obtained for the RF-CAP. That is because this technology, the self-resonance coaxial cables, they don't couple so much with each other. And we find out that our RF-CAP actually outperforms the 32-channel coil or the 20-channel coil. I mean, performs way better than the 20-channel coil, but it really outperforms the 32-channel coil in one direction, when we use only one direction in this case, but also when we do it in two directions, the acquisition, which is really very interesting. I believe it's both because the correlation matrix is so low and also because we are like closer to the head. So, but that's very important because, as I said before, if we want to reduce those distortions, the geometrical distortions, we want to speed up the acquisition, and also we want to have a better time resolution. So now let's talk about interactions. What happens if I place the TMS on this resonant element? Because those elements are resonant to the Lamont frequency at the 3 Tesla. So first of all, they're going to be detuned a little bit, in some cases a lot. So it depends a little bit. We wanted to assess that. And then we're going to have also some kind of, what I call it, the eddy currents effects on the V1 minus. And if you are really interested in that effect, you can read it from our publication. If you place a TMS, for example, you have your RF coil here. Imagine you use a flexible coil here, and then you have the bring here. You're going to have some kind of shielding of the signal. You're going to have very low signal under the TMS. If you put it under it, the RF element, it's way better. We still have that effect, but way less. So I wanted to do an assessment of that effect. So that's why I measure without the TMS and with the TMS, and this is a relative SNR. So this means how much of the SNR I am losing when I place the TMS. And you can see that the effect is about 10% what we have in the brain when we have the TMS present, which is, you know, it's nice to have this kind of inform to the technology so that we know we have probably 10% less SNR, which in the cases of the cortex where we have such a high SNR is not going to be a big deal. So in terms of the TMS efficacy, as I said before, because the TMS, it's very important to lie really directly on the scalp. We're going to have some effects on the stimulation intensity that you need. So for that, we wanted to evaluate in 3 subjects that effect. And for that, we determined the resting motor threshold. So the resting motor threshold is the intensity that you need in your stimulator so that you produce, when you place it over the motor cortex, that you produce only half of the time a muscle twitch. Or if you are measuring everything with an electromyographic system, that it has to be at least 50 microvolt peak to peak. So we determined the resting motor threshold without the cap and with the cap. And what we realize, of course, it increases it. We need a little bit more because we are losing due to that distance, but it's between 6 to 8% of that MSO. We also did this kind of evaluation, in this case, only on one subject. We are still working because our final destination is actually to integrate also the EEG. We wanted to see, okay, when we place this EEG cap, the EEG electrodes are a little bit thicker. What's going to be the effect? And in that case, we saw that we are losing about 11% of the MSO, 11% of the intensity. So with all these things clear, okay. Yeah, so I think we have shown also the usability. So even though you are losing some of the efficacy of the magnetic field, we still have a lot of room to have experiments in the scanner without losing, I don't know, 30% of the stimulation, like it wasn't the case in some cases. So now, after all this technical data and everything, I want to show you some of the in vivo data. So this is the structural images that we acquired with the RF-CAP. And then we have the multiband, the functional images, the 2D API with a repetition time of 2 seconds. This is an isotropic box, it's 2 millimeters, with a multiband of 2. But it's not only nice images what I want to show you. I want to show you real results also from an experiment that we have done, that we have started now with our RF-CAP. And of course, we have to start with the motor cortex because it's the best studied right now and the easiest to control. So our methods or our experiment consisted of two MR sessions. The first MR session, we had a functional localizer we calculated a functional localizer using a finger tapping paradigm. And then we used the finger tapping paradigm or that functional localizer for the second session, so that we would use the neuronavigation to place the TMS over the RF cap so that we were sure that we were, on the right spot. And with the RF cap, we determined the resting motor threshold, the way I told you before, with the EMG. And this MR-safe EMG is going to stay in the scanner the whole time. Then we did the stimulation protocol. So we did actually the second session started with structural images and then some images to correct the distortion, but then the stimulation protocol, the TMS-fMRI, always with the EMG in the scanner. So our stimulation protocol lasted 3 minutes. It consisted of 5 blocks, and we have in that block always a 8 seconds stimulation block that is shown here, and then 28 seconds with no stimulation at all. And each of those TMS stimulation blocks are in an interleaf way, as I said before. So the volume acquisition took about 623 milliseconds, and then we place here not a single pulse, but a burst of pulses. And I will tell you, we switch from 1 hertz, because this TR is usually 1. We wanted to switch from 1 hertz to a burst stimulation of 20 hertz, a little bit to push the hemodynamic response and to see some cumulative effects. We knew that we were going to see some cumulative effects. That's why we lowered the intensity to 50, 65, and 80% of the RMT. Then we did also single pulse with 110% to compare it nicely with the finger tapping. We wanted to see, is that, if you stimulate only 1 second and we ask the subject to do 1 second more or less finger tapping, is that comparable? We did that also with single pulse. And then this is the way we acquire our functional images with 2D single shot DPI, TR of 1 second, T of 30 milliseconds. We use multiband 5. We did implant acceleration 2 so that we could achieve 2.5 millimeters isotropic resolution. We wanted to cover even the cerebellum. That's why we push it so much. We left 100 milliseconds margin for this interleaf so that we were sure that we are not introducing any artifacts in our images. And then we did the data processing with FSL. We only did the motion correction with AFNI, but the rest with FSL. And now, these are single subjects with result, what I'm going to show you. So we are now collecting more data, but unfortunately, I only have single subject results, but I think they are very nice, and I want to share them with you. So we did always 2 runs for the stimulation, and for the finger tapping, we did 4 runs. So now we're going to focus on reproducibility. This is the first run of the stimulation with 50% intensity, and then this is these results that I'm showing you is with no smoothing at all. So we keep the 2.5 millimeters resolution. And then this is the second run. Now here in this picture, I'm showing you the overlap, and we calculated also the dice score so that you can see how spatially constrained or similar it is, even though we have in some cases, especially in the auditory cortex, a little bit differences. In the run one is in red, run 2 is blue, and the overlap is in pink. These are the results for the 65% RMT, a little bit more recruitment, more activation in the first run. This is the second run, and this is the overlap of that. In this case, the dice score went up to 0.559, so almost 60% of the activation is the same. And again, in red, the run 1, blue, red, run 2, and the overlap in pink. Then we have, for example, for the single pulse, stimulation. This is the first run. Then we have the second run. I want to say that the 110 is going to probably move a little bit too because it's higher intensity. But I mean, the dice score went a little bit down, but it's still even better than the 50%. And I don't know how familiar you are with the dice scores. I am not, I have to be honest. It was something that I have just learned a little bit. But I wanted to see, okay, how are we actually with the regular finger tapping with the fMRI? And so I took a look at the runs that we have for the finger tapping. So that is the run 2, that is the run 3. And actually, we have even a dice score that is a little bit even worse than the ones that we obtained. And I told you before, we had 4 runs. So our dice scores range from 0.33 to 0.44. So I'm showing you, of course, the best, but that you know that the fMRI is already a little bit limited on the reproducibility that we can have. There's, of course, we are measuring also biological systems, so we cannot expect to have 100%. But from what it looks, at least for this single subject results, is that introducing the TMS has not have an effect on the reproducibility the other way around. Maybe we are controlling better our stimulation, we are controlling more, or we have a more control experiment, and maybe that's the reason why our dye scores. But of course, these are all hypotheses. These are just single subject experiments, but it's something to keep in mind for the future. So now I want to talk about precision. So what I'm showing you here are the results from Bestman et al. In 2004. In my opinion, the best concurrent TMS-fMRI over motor cortex full brain coverage that has been done. So his stimulation protocol consisted of 5 minutes. And what he delivered is he had also 9 seconds stimulation and then 23 seconds off, very similar to us. But in his case, he had to deliver those TMS interlace or interleave with the slices. Okay, he couldn't do the fast acquisitions and everything. So he had a 3.3 hertz stimulation. So he delivered in total 30 pulses per stimulation block and a total of 240 pulses. And this is what he found out. So he reported he did everything in two stimulations, 90 and 100%, 110%. So he reported that they could only see activation under the coil for the suprathreshold stimulation and not for the under threshold. They also have the EMG in the scanner, so they could show that the 90% did not produce any movement. So the hypothesis was that maybe what we are seeing here is the afferent path. That's it. We are seeing that the finger moves, but not that we are activating, not the activation from the TMS, which is strange because they were reporting activation, so both activation from the under threshold, for subthreshold. So that was always the discussion in the community. What is going on? I mean, in my opinion, it was always that they just had horrible distortions or signal drop-offs, or it was just not good enough. Another point is that the resolution that they have a temporal resolution of 3.3 seconds, and it's a little bit difficult to know exactly the voxel sizes because they are quiet with 2 by 2 by 4, but then they interpolated to 3 by 3 by 3, and there's no information if they did some smoothing, so it's difficult to compare it, but I'm going to just try it. This is our stimulation protocol. As I said, our stimulation protocol is 3 minutes only, not 5 minutes. We have per stimulation block, we have a similar number of pulses. We have 32. Ours is all in burst. It's not that 3.3 hertz. And then we have a total of 160 pulses in that case. In the case of the single pulses, we reduce it even to 40 pulses only in the whole. So these are our results. And you can see that we see stimulation under the coil. We see that one, and we have also EMG in the scanner, and we see that there is no movement. So that stimulation that we see there must come from the TMS coil. Of course, very, very careful. This is single subject. We are working now on that. But I hope we can show, at least, resolve this kind of mystery. So this is what I was promising you, the EMG. These are the average epochs of the stimulation. You see here, these are the TMS artifacts, actually, because we give it in burst. For the single pulse, you have only one of those artifacts. And then you see there, and the part starts to be the gradient artifacts. They are very, very impressive in the EMG, because EMG is just like the EEG. It's going to have, you're going to induce high voltages there. And then you start to see in the 65% already, and the 110 is very, very clear, movements of the hand here. And this is, as I said, an average. That's why you see it like that. And in the moment that the hand moves, because the cables also move, the EMG starts to go a little bit crazy. Especially for the 110, it's very nice to see that you have that first motor evoked potential there, exactly 20 milliseconds from the TMS artifacts, and then the cables start to move a lot, and that's what it's collected. But you see that there is no movement for the 50% RMP. So, okay, oops, where did it go? So fast. Okay. So now let's talk about reliability, the signal that we obtain. Okay. So here from that experiment, also the signal time courses. This is average over all the stimulation blocks and over all their participants. They have, I think, 5 or 6 participants. So they have, for the finger tapping, not even a 2% signal pulse change. For the 110, the one that they reported as activity, that is on the left M1, sorry. They have not even a 1%, and then the 90% didn't show any significance. If we compare that to the data that we have, again, single subjects, but it's still very nice to see that we see the ball response and percentage ball change nicely for all the intensities, and they are correlated with, I mean, the level is correlated with the intensity. And even with the single pulse, we also see a nice, almost a 6% ball change. So these data give us more confidence that there is something there. There's a hemodynamic response. There's some oxygenation there. So with this, I hope I could convince you that with the RFCAP, we are going to improve the measurability, the precision, the reproducibility and reliability, and also the usability of it. So now, with the RFCAP, I think we can push the measurability, as I said before, at least to a 9. And then the reliability and reproducibility, a very big topic or important topic or difficult to solve, to a 7. And then to accessibility and usability, I will even dare to go up to 8, because, I mean, we've done these experiments. I've done these experiments before with the disc coil, and they were a nightmare. They were very difficult to position the coil, especially, was a nightmare. Very, very difficult. Now with the cap if Kenny is here, my witness, it went way easier. And we still don't have the perfect fixation system, but it's still very, very improved a lot, definitely. And also with showing that we are not having such a big effect on the signal is also improving the validation. So now let's talk about other methods. I don't know how I'm in the time. Yeah, so other methods like to improve the calibration and reproducibility. So I'm going to be talking of the near real-time e-field modeling. Actually, this is not my work. This is the work from Mohammad Daneshan, that is not here, but of course of Apple. But let's start with some concepts of TMS quantification. So we have the targeting, that is where we want to have the desired brain location or stimulation. And then we have the dosing, which is the strength of that stimulation. And the dosing is going to depend on 3 factors. First is the pulse strength, the current that we are going to apply to that, to the TMS coil, but also on the stimulation pattern, that if it's a burst or the frequency that we are going to give it, and the duration of that. So with the computational models that exist nowadays, and Apple here has been leading the development of the BMFMM, you can really quantify the spatial pattern and the amplitude of that electric field that you are inducing. You can also use other software packages. But we know that, for example, the effect for the dosing, the effect of the stimulus patterns or the duration is more challenging. And that's why we need really concurrent TMS-fMRI. And you could see it in the results that I was showing you, that with single pulse, we have a 4%, even though it's higher intensity, very similar to the 50% when we have a burst. So there is some information there that we can really only see with the concurrent TMS-fMRI. So for the computational modeling, so we can really estimate the electric field distribution using these computational models. And most of you know that the maximum is going to be under the coil, and it's clear. So if we have this figure of 8 coil and we have those currents in that direction, the maximum is going to be there. And if we calculate it, it's really there. So it's a maximum field, in this case, of 68 volt per meter. So why do we really need a more quantitative approach if we know that the highest is there? So let's take a look at some examples to understand why it's important. For example, the effect of the coil location. Let's assume we have the stimulator output, the intensity is the same, and then we move the coil between those 2 positions. One is this position, and then we move the coil to this other position. What is the E-field intensity? Do we expect that it's going to be the same in the brain? So then we calculate it, and then we calculate this is 68 volt per meter, and this is 59 volt per meter. And why is this? Well, the brain is modifying or is going to shape the E-field that we are inducing. So that is well known. So that's why it's so important to do it. measurements. And it's really the difference in E field, in this case, that is over 10%. When we are doing these motor cortex experiments, is it 100% the same as 110? No, 100% is going to produce only half of the time as muscle twitch. With the other ones, we will have most of the time, so at least 8 times. So there is a difference. So we should not just ignore that. Then in the case of the rotation, if we have the same location, but we rotate 90 degrees the coil, for example, we have this position, and then we change it to this position. Well, the amplitude is the same, but the shape and the direction changes. Especially you can see that, that direction means that we are going to hit or we are going to activate different neural populations. So that's also an important topic. In the case of a tilt, very similar. We have the same location is fixed, and then we just tilt it in the left-right direction by 20 degrees. The effects is that in this case, both changes. We have changes in the amplitude, but we have changes also in the spatial distribution. So that means we need to control or to know what is happening when we are placing the TMS coil and talk about volt per meter instead of resting motor threshold percentage or something like that, so that we can have a better calibration. So for that, I mean, you know, showing the work from Mohammed, this is the near real-time E-field modeling. This is individualized for each patient different. So it's based on the theory of dipole-based expansion. So what we have is a pre-calculation step. So we need MR images from the subject to do a good segmentation. And what we are going to do is we cover with set of dipoles, the head with the nerves, and we pre-calculate those E-fields for each of those dipoles. And then when the subject comes in and we are doing the neuronavigation, then the E-field of that moving coil is going to be just the interpolation from those stationary dipoles. So that the field position, the position of the coil is going to be tracked by the neuronavigation, and the total field can be calculated. And I'm going to show you a video that unfortunately it's not working here, but I hope I can show it like this. Yeah, okay. So now, so you can see that it really, this the first element is keeping track of the coil, and the other computer is calculating that E-field near real time. So now let's go back to the, okay. And this is the right one, yeah. So I do believe with this near real field calculation, we can improve also the reproducibility, and of course, we can do something to have more calibrated measurements. So now, adding this E-field modeling, as I said, I think we can push a little bit the reliability and reproducibility, and we can do something in the terms of calibration and standardization. So I just want to see how we have pushed everything, or I hope we can push that in this direction so that the concurrent TMS-fMRI becomes a more reliable and scientific tool. And now, just to wrap it up, so from the day it was born, the 1998, as an extension of human perception, I think a lot of work has been done by many people to, work on the measurability, on the validation in the 2000s and the end of the 2000s with fixation systems, with leakage current compensation, and so on. Then with the work that I did in Vienna, we also pushed a little bit the measurability. It's still challenging how to handle everything. They have been doing a lot of, extending that with 2 elements, and then the near real field calculations can help really with what I think is one of the weakest points, is the calibration and standardization. But I also think that the RF-CAP is a key component that is going to enable the transition of concurrent TMS-fMRI into a robust scientific tool. And now, before I go to my last slide, I want to present a very, very interesting event that is going to happen in Vienna in April. So it's the second workshop of MRI of neuromodulation. I'm very, very involved in the organization of this workshop. So we just started the call for abstracts, and we are going to open soon the registration. So I really invite you. We have great speakers, keynote speakers, very great confirmed speakers. I hope Apo too. And okay, and with this, I just want to thank the funding mechanisms, the collaborators, especially Jason Stockman, who has been always incredibly supportive, and of course, Apo and the whole group of the TMS core, who, gave me the possibility to evolve all these ideas. And of course, thank you for your attention.